Hello, welcome to our fourth lab presentation, if I'm not mistaken, on enzymes. And this is done by Ashley Patel and Stacey Brennish, just as a side note. So, like I said, this is on enzymes. And let's start off with asking ourselves, what is an enzyme? So, an enzyme is a globular protein that catalyzes chemical reactions. So, essentially what an enzyme does is it speeds up a chemical reaction by lowering the activation energy required to carry out a chemical reaction. And in this experiment, what we're trying to figure out or establish is what is an enzyme's prime temperature or pH. And, how does having the prime temperature or pH affect its function? How does it better it? How does it lessen it? And the enzyme we're using, because we have such a wide variety of enzymes and each one differs in its temperature and pH, the enzyme we're using is catechol oxidase. Now, for those of you who don't know what catechol oxidase is, it's that enzyme in our fruits that turn that delicious color brown that makes that fruit so appetizing to eat afterwards. I'm joking. But, yes, this enzyme, when it comes in contact with air, once the skin of the fruit is peeled off, we know that it turns brown because it has an end product that has that color. So that is the enzyme that we're using today. Sorry, I got a little lengthy there. So our hypothesis, what do we think about all this? What do we think is going to be the prime temperature, prime pH? So based on our somewhat knowledgeable minds, we decided that, most likely the proper conditions should be around our body temperatures and maybe the pH of our blood. So 35 degrees Celsius, close to 95 degrees Fahrenheit, and a pH of 7, where our blood is at a pH of 7.3. So we figured those are pretty good numbers to go by. So our experiment. Now, when we started the experiment, the first thing that we wanted to know is, what is a positive response? What does it look like? How do we know that we have a positive response? And so we established our controls for temperature. And after we established our controls, we proceeded to test different temperatures and different pHs to see which ones worked and which ones didn't, which ones had a result, which ones slowed down the function, and all that fun stuff. So what we're looking at here in this picture is our three controls, and we're looking at two negative responses and the positive response. So our first test tube on the right, because this is backwards for some reason, our first test tube on the right is a positive. 2 and 3 are negatives. And these test tubes, they all had 1 ml of water, 1 ml of enzyme, and they were all placed in room temperature water, which is about 37 degrees Celsius. And no, it's a bit harder. But these test tubes were placed into 37 degrees Celsius water, and they were left there for 10 minutes to heat up the enzyme, and then when they were pulled out, we added catechol oxidase, not catechol oxidase, just catechol itself, to them, and we waited to see if there was any visual reactions, and this is what we saw. We saw I apologize. I just told you a lie. In the first test tube, we added catechol. In the second one and the third one, we didn't, which is why they become negative controls. Otherwise, they'd all be brown. And I just fumbled so badly, and I apologize for that. So, first part, or part A of our experiment, is temperature. And we had to come up with an experiment. So what we did was we put 1 ml of catechol oxidase and 1 ml of water in 6 individual test tubes labeled 1 through 6. And each test tube had its own water bath. When we say water bath, we soak them, or we place them in water of a certain temperature and allow them to heat up for 10 minutes. So test tube 1 was in 40-degree water, test tube 2 was in 60-degree water, test tube 3 was in 100-degree water, test tube 4 was in 80-degree water, test tube 5 was in 10-degree water. This also was an ice bath. And test tube 6 was just kept at normal room temperature water. And after 10 minutes, we took the enzyme at its new temperature and we added catechol to each test tube. And we measured their reaction. So this is our control. This is the before picture, what we had before we knew what the change would be. This is right after their water baths. And you might be wondering why there's a gap. It's because test tube 5 is off in the corner right now because it took a while to cool up. So that's test tube 5. And what we did is we added catechol to each one. And voila, here are our results. We find that the test tube 1 in 40 degrees did really well. It got a shade of 3 in darkness. Oh, I forgot to tell you guys, we're basing these from 0 to 3. 0 meaning no change, 3 meaning a lot of change. So test tube 1 is a 3. Test tube 2 was at 60 degrees Celsius, and we leveled that one as a 3 too. Test tube 3 was at 100 degrees Celsius, and we can tell that it's clear, there's not a lot of change. And what happened was, because of such a high temperature, the protein denatured, and the enzyme lost its structure, therefore it could not do its job. So there was no reaction to the catechol. Test tube 4 was at 80 degrees Celsius, and there was a definite change there. So we labeled that as a 3. And test tube 6, which was our control, again, was a 3. It was in room temperature, about 37 degrees Celsius. And that's test tube 5, which is our ice water bath. And what we found was that it didn't have much change. And this was because when a protein is cooled or nearly frozen, like we did it, the production of it slows down and sometimes even stops working. So that's our temperature portion of the experiment. Now, our second portion of the experiment was our pH. What is a prime pH for the enzyme? So, our experimental design, it started off the same way. Place 1 ml of catechol oxidase and 1 ml of water into individual test tubes, and these test tubes are labeled 1 through 8, and then we're going to add 1 ml of every pH that we had in every test tube. So the first test tube, pH 2, second test tube, pH 4, third test tube, pH 6, fourth test tube, pH 7, fifth test tube, pH 8, sixth test tube, pH 10, seventh test tube, pH 12, eighth test tube didn't have any pH. And after the pH sat there for about 5 minutes, we put 1 ml of catechol into each test tube to find the results. And this is our before picture. They're in order, and they're looking kind of cool, just chilling there. And after about 3 minutes, 4 minutes, I believe this is our picture, after about a good 15 minutes, we started seeing changes. And what we saw was that the pH 2 had no reaction, 1. The second, pH 4, second test tube, again, no reaction, very, very, very light change, so barely anything. We labeled it a 1. pH 6 showed a reaction. pH 6 through pH 10 all showed a very positive reaction, all labeled 3. All differed in their time getting there. However, in the end, we can tell that they're all about the same. pH 12 had a slighter reaction. We labeled that a 2. And our pH 8 is a very light. So again, we labeled that as a 2 going to a 3, but we stuck around 2 because it didn't have such a striking color as the others. So our conclusion. What did we learn from all this? What do we know now for catechol oxidase? So what we found was that if a temperature of an enzyme is too high, it will denature. It will lose its structure, and as we know, structure if its function. And when this happens, it can no longer carry out its function. If a temperature of an enzyme is too low, then the function slows, and again, this impacts its ability to, move forward and actually carry out the reaction necessary. If the pH is too basic or too acidic, what we found was that the structure of the enzyme and the protein itself is affected. And again, structure fits function, and it's completely messed up. And for a teenage word, it gets messed up, and now the function and the end product is affected, and there is no reaction. So, were we right? Were we wrong? Our hypothesis said that at 35 degrees Celsius, the protein would function at its best. Our results showed that the enzyme functioned well from temperatures to 40 degrees Celsius to 80 degrees Celsius. Those were its best reactions, its best, most brownish color. Sounds so bad. But based on the pictures that we saw, we know that this was when it reacted the best. We also hypothesized that the best pH for an enzyme was 7. And we ended up finding out that the enzyme could function well at a range of pH 6 through 10, which was a bit unexpected because of the range that we thought could happen. But like we said, we learned this. So now, sorry, I clicked the wrong button. This brings me to the end of our Prezi sad face, because I actually really enjoyed doing this one. But I hope you guys enjoyed it. I hope you guys liked it. I hope you guys, learned something new, because we sure did. So, buon provecho.